My name, for those of you who don't know me, I'm Pagnucco. I'm another PhD student. And I'm very honored to introduce your 2015 McGill Biology Department student speaker, Monica Granados. I was very honored to be asked to introduce her. Monica is a very, very dear friend of mine, and I will try not to embarrass her too much as I go through some details I know about Monica. Before we get to the present, Monica came to us by way of the University of Toronto, where she did her bachelor's degree and a master's under the supervision of Don Jackson, where she was looking at detecting changes in fish communities in response to habitat rehabilitation. She came to McGill in 2010, where she has left an indelible mark on the department. Her thesis with Gregor Fussman is looking at interaction strengths and the influence of omnivory on food webs. And her thesis has brought her to international conferences, and in addition she was invited to Carleton to give a talk on her research. But where Monica's really shined is in establishing collaborations with students both at this university and beyond. One of the highlights is a paper she did with two other PhD students from McGill, Etienne and Corey, on the rising complexity and falling explanatory power in ecology, which was published in Frontiers. I also had the pleasure of collaborating with Monica on a project she'll be talking about today. We've also collaborated on many other projects you won't hear about. In particular, several novelty papers that we co-wrote that were inspired by terrible punny titles. I'll give you one example. What's good for the goose is good for the sander, a newly discovered mutualism between the Canada goose and walleye, Sander avitrius. Monica's strengths go well beyond science. One of her key abilities is to blend science and design, in which anyone who's ever seen her give a talk can attest to, or saw the fact that she created a GIF in lieu of an abstract for this very talk, which is ridiculous and amazing. She also has a blog about how she integrates design and science called Science on Tumblr. You should check it out. Lots of great tips on different ways to create cool visual tricks in R. But especially Monica has given back tons in the form of public service and outreach. Many of you who've been here long enough will know that Monica served on the BGSA executive for several years, including 2 terms, I believe, as president, which is no small undertaking, as I'm sure Mary Rose could also attest to. And she also does tons of volunteer work, where she gives her time to analyze data for nonprofits, such as Data for Good Montreal. And she also volunteers in Toronto at Action Potential Lab, which combines science and art in this fun lab atmosphere for kids and adults. So Monica has numerous gifts, and she's very generous to share with them. I'm sure many of us have gone to her from time to time to get some R-Stats help, or a poster that we need for something, or DJing for a party. Monica is the sort of multi-talented, generous, brilliant scientist I think every department wants and needs, and she'll be bringing her gifts to Université de Montréal next, where she'll be doing a postdoc with Dr. Timothy Poisseau, developing a model to infer trophic relationships based on size structure in communities. Very quickly, please head with us to Thompson House after the presentation, where we'll be celebrating Monica and this beautiful weather with beer and nachos, the best way to celebrate, immediately after this seminar. So without further ado, please join me in congratulating and welcoming our 2015 student-nominated McGill Biology Department student seminar speaker, Monica Granados.
Thank you for that lovely introduction and for omitting embarrassing details of our friendship. So I am Monica Granados. I'm going to be talking a little bit about food webs and omnivory in particular. So I've spent the last 5 years studying one configuration that you find very frequently in natural ecosystems. But the twist of what I was interested in was finding out given that this module has been studied so much throughout the history of ecology and in the field of ecology, there are a lot of predictions that have come forth and have arisen as a function of studying this food web configuration. But if we were to introduce this food web configuration to a novel ecosystem, would we find that the predictions that have arisen through empirical work and through theoretical work, would we find them to be consistent in this context? So my talk is going to be about that, about testing the predictions of omnivory in food webs using introduced species. And then at the end, I will also talk about how I'm extending these ideas to my postdoc work in the future. So I think the general public likes to believe that when they think about consumptive interactions between a predator and a resource, that it's very linear. You've got this one big animal that eats a smaller animal, and then that smaller animal even eats an even smaller animal. So you learn about the food chain when you're growing up in the 3rd grade, but then you become an ecologist and you realize that that configuration is actually not consistent. You find that in real food webs, you find that omnivory is really prolific. So omnivory, in the context of this talk, is feeding on multiple trophic levels. So you've got that big animal that's eating that smaller animal, and then that smaller animal is still eating that common resource, but you've got that big animal that will also eat that basal resource. So it's feeding on multiple trophic levels. So it turns out that if you go out to real ecosystems, you don't find that linear food chain, but rather you find that omnivory, so this feeding on multiple trophic levels, that connection between that top trophic level to that common resource is really prolific. You find that between these consumer resource interactions, there is that connection between the predator and the common resource. In fact, looking at some real food webs that have been mapped out, Arim and Marquet found that between 54 and 87% of the food webs that they looked at, that were mapped, had some kind of omnivorous interaction. And so that's a pretty high number for a field as variable as ecology. Thompson did a similar study where he was looking at, or him and his colleagues were looking at how often do we find that predators are feeding on multiple trophic levels. So what they did was look at, try to quantify how many different trophic levels are they eating on. So they were trying to quantify how many times are they eating on just one trophic level by determining whether the average number of prey that they were eating were on the same trophic level. So whether or not they were integers or not. So every trophic level got an integer. If you fed on multiple trophic levels, then you had a non-integer result. And they found that marine food webs had the most amount of omnivory, so all the way up to 60% of the food webs had non-integer trophic positions. And above herbivores, that most of them were omnivores. So they had this null model where they were testing what's the likelihood that you would get this distribution of non-integer values, and they found that there was no difference between that and chance. So it was more likely that these interactions were sort of this mix of, he called it an omnivorous tangle in these food webs. So that raises the question, so why is omnivory so prolific? Why do we find these omnivorous connections so frequently in food webs? So one of the main reasons why is this ability of the predator to be able to eat two different prey types. So because it can eat these two different prey types, it can adapt its feeding to feed on the higher density resource. So in the event that the common resource is low, it can increase its feeding on the consumer, here, snails. If the snail density decreases because it's eating a lot of the snails, so that interaction is high, then it can switch to eating more of that common resource. And so that switching back and forth essentially allows that resource to bounce back. It doesn't drive it down to extinction. So allowing it to switch back and forth means that that configuration or that food web module is more stable because the constituents are persisting. couple of empirical tests of this this configuration, or these predictions of the configuration. So there was an experiment by Fagan in 1997, and what they did was look at different types of communities that they built. So they built some communities that had more omnivores, and some that had just predators, and some that had sort of like a mix. And then what they did is they applied some kind of a stressor. In this case, it was an aphicide. So they essentially killed off a lot of the consumer population and tried to see what happened to the levels of prey or consumers in these little microcosms that they had done near Mount St. Helens. And actually what they found was that those communities that had more omnivores were more stable, and that they had greater numbers of prey after the application of the aphicide. Probably because of the mechanism that I had explained earlier. So allowing it to feed on multiple different types of prey means that you're not concentrating on just eating one particular prey type and driving that to extinction even after a stressful event. And then in 1998, Julio Kinshashtev did another cool experiment where they again built these communities, but this time in microcosms. And what they did is they had a gradient of omnivory. So they chose species that were more omnivorous or less omnivorous and created a gradient so that they could explore, as you increase the number of omnivores, or at least the omnivorous feeding in these microcosms, did that confer that stability or that persistence that is theorized or predicted by food web theory? And they actually did find that persistence increased as you had more omnivores. So if you had more omnivorous predators, you had greater persistence. But all of this only makes sense in the context of interaction strength. So interaction strength is the concept of what's the likelihood of your consumer or your predator eating a resource or your prey. And for omnivory to be stabilizing, so for that switching to work, you have to have interaction strength be weak. And that's a lot of the work that Kevin McCann has been doing, looking at how interaction strength mediates the stability of food webs, and particularly in omnivory. And what he found was that interaction strength has to be weak in order for omnivory to confer this stability or the persistence. And that's because if you have an interaction strength that's too strong, then you'll drive that consumer or that prey type to extinction, even if you switch back and forth. So if that's too strong, you don't allow the prey or the resource to bounce back up. And so a lot of the work that I'm going to be looking at is, do the predictions of omnivory, are they consistent in these introduced communities, and do they depend on interaction strength? So the question is, but if we introduce omnivory, will it be still stabilizing? Does it promote persistence in the context of introductions? We know it does in these natural communities. I've shown examples where that's true. But if you introduce it into a novel community, will it still work the way that we've seen it work in these natural communities? So the first experiment I want to talk about is, it's a meta-analysis where I was looking at published literature on invasive species. So I looked at about 22,000 papers looking for examples where omnivores have been introduced, to look at what are the impacts of this omnivore being introduced. And I wanted to compare it to something so that I had an example, I had some kind of quantitative analysis of what's the impact of an omnivore, introducing an omnivore versus introducing some other, any other type of feeding guild. And so I looked for papers that had introduction of an omnivore and an introduction of a predator. So given what we discussed earlier, you would expect that omnivory should have less impact because it has access to more resources, right? So the impact on one particular community should be less because it could switch back and forth. So how did I go about doing this? As mentioned, I went through 22,000 papers looking for specific papers that fell under certain restrictions that I placed in order to minimize the variation between these. Because when you do a meta-analysis, you want to try to maintain the variability between all your papers so that you can ascribe your results to just the differences between the two focus groups that I was looking at, so omnivores and predators. So I selected that papers have to have two rhesus-worthy omnivores, so the omnivore in the experiments that was published had to have not only an effect on the consumer, but also then on that common resource. You had to have a control, so the papers not only had to show what the impact here being like how many individuals of the prey type were present in the presence of an introduced species and in the absence of the introduced species. Okay, so a lot of these were, some of these were field experiments, so there would be a field experiment where they had these exclosures, so they would see what's the impact of having a crayfish in this enclosure versus having it outside of the enclosure, like what's the impact on its prey resource, so like snails or invertebrates. So I was looking at how those levels of the invertebrates or phytoplankton changed in the presence of this invasive species and in the absence of this introduced species. And then I had the added factor of if it was a predator or if it was an omnivore. But as we discussed earlier, also this impact depends on interaction strength. So we might get more information about what's happening to these communities and what this effect is if we know a little bit about the interaction strength between the predator and its resource. So how do we go about doing that when we're not actually doing an experiment, when I'm doing a meta-analysis looking through published literature? So what I decided to do is to look at functional response. So functional response is something that's come into the foray of introduced species or invasive species because there seems to be a really interesting correlation between the functional response of a species and its impact. So that it seems that a lot of introduced species have a stronger functional response than native species, potentially then indicating a mechanism for why some introduced species have such a big impact on native communities. So what's functional response? So a functional response experiment, what happens is that you do different experiments at different prey densities. So you've got a prey density of, say, let's say, 0, 1, 3, 4, and 5 in separate receptacles, say like beakers. And then you look at how many of those prey are being consumed by the predator. And so depending on the shape of this curve, you can know a lot about how that predator is consuming. So what's the attack rate? What's the handling time? And that information can give you a lot of information about this interaction strength. So what I did was use it as a proxy for interaction strength. So using these two pieces of information, so the standardized mean difference was what I got from the papers showing impacts in the field or in the laboratory. So here I was looking for what's the mean abundance or density when the introduced species was present versus when it was absent, and then it's divided by a measure of variance to weigh the different studies by their sample size. And then I used the Rogers-Predator equation on that functional response to pull out some interesting parameters that I could use as a proxy for this interaction strength. So I pulled out the attack rate and the handling time because that gives me some kind of idea of this interaction strength. So a higher attack rate is a proxy for a stronger interaction strength. So what did I find after going through these papers? Interestingly, and contrary to what I had expected going into the analysis, actually found that omnivores had a greater negative impact than predators. So on this axis, is standardized mean difference, meaning that as you get closer to 0, there was no difference between the predator being there or not being there, or the introduced species being there or not being there. And as you go positive, it means that there was a beneficial effect of having that introduced species there. As you go to the left of the 0, you have a negative impact. And so I found that there was actually a greater negative impact of omnivores than predators. And so that was interesting and peculiar because I initially expected that the omnivores would have less of an impact, right? Because it's able to switch back and forth between prey type. And so that's when I moved to looking at this attack rate, or looking at a measure of this interaction strength. So here I'm plotting effect size, so what the effect was on the recipient community, and on the x-axis I have attack rate. Here, all my predators are in gray, and all of my omnivores are in black. And as you can see, if we're looking at just the x-axis, the attack rate is higher in many of those omnivores. So a lot of these omnivore species have greater attack rates than the predators that I used in this analysis, this meta-analysis. So likely then, the reason that we found that difference between predators and omnivores, especially contrary to what I had predicted or anticipated, was that these attack rates that are stronger are driving these lower or greater impacts or lower densities in the presence of these introduced species. And interestingly, we did find a pattern of, as your attack rate increases, your effect size also decreases. So the greater attack rate you have, the more negative effect you're going to have on that recipient community, which is what the group that's working on these functional responses predicted. So they predict that if you have a stronger functional response, you're going to have a stronger impact, but it hadn't been mapped out because we still sort of are amassing different measures of functional response because it's a relatively new field. So this is sort of like the first go at looking at the relationship between effect size or impact and attack rate. So here, omnivores had a greater negative impact due to stronger interaction strengths. So although the predictions weren't consistent with what you'd find in native communities, it is consistent with the predictions of the theory of omnivory because it's dependent on these interaction strengths. So because the interaction strengths were strong, then we found that there was a bigger effect by the trophic group that had stronger interaction strengths. So in fact, the results did corroborate the theoretical predictions about omnivory in this case. So what about stability? So in this case, I was sort of just looking at interaction strength and impacts, but I wasn't looking at stability. So what if we introduce an omnivore? Does that, if the omnivore is introduced, does it promote stability? So as Katie mentioned, we got to work on this really awesome project looking at the effects of origin and omnivory on stability. And here we used a system, a mesocosm system, consisting of crayfish, snails, and then benthic algae. So that is our system and our configuration. But what makes this experiment particularly neat was that these are the mouths of two different crayfish. And so crayfish are naturally omnivores. So they feed on snails using their chelae, and they also feed on benthic algae using the setae that they have in their mouths. So what we decided to do was, in order to determine what the effect of an omnivore is, very similar to the meta-analysis, we needed some kind of comparative module. We needed a predator. Omnivores aren't predators. So we decided to make them predators by giving them haircuts, as I like to affectionately call it. And we removed the setae from the mouthpieces that traps the benthic algae in their mouths. So essentially, we removed the ability for them to eat this benthic algae by giving them these haircuts. So that resulted in these configurations. So we had an omnivore that could eat the benthic algae and the snails, and we had a predator that could just eat the snails, and then the snails in turn ate the benthic algae, and same thing in this case, right? So we recreated these two different modules. And then what we did was, so we replicated it, we let it run for 60 days. We measured stability by looking at the density of benthic algae in these tiles. So every 2 days, we remove a quadrat from these plexiglass squares or tiles that we had set at the bottom of the mesocosm, and then we mapped how that density changed over time. And that could give us an indication of stability, because if that variation is lower, then we know that that is a measure of stability. So what did we find? We found actually that, as predicted, omnivores, here in black again, had a lower coefficient of variation. So having those omnivorous crayfish, regardless of the species that we used. So if we, in the experiments, so not only did we have predators and omnivores, but we also had native and non-native species to look at that aspect of introduction. So regardless of if it was a native or non-native species or an omnivore, we found that, omnivores conferred a lower coefficient of variation. We also found that there was actually some species differences that sort of ran across the native versus non-native. So here, rusticus and lamosus are two non-native species, and Oncorhynchus virilis and propinquus are native species. And as you can see, we actually found that rusticus and virilis had lower coefficient of variation relative to these other two species, and that actually turns out was driven by interaction strength. So these two species ate a lot more of the snails than these two species. So the patterns were again consistent with this idea of the predictions of omnivory, or the predictions of these consumptive interactions, depends a lot on these interaction strengths. So where we had strong enough interaction strength to suppress the snail populations, we had a coefficient of variation. In this case, the interaction strength was so weak that it was almost like the predator or the omnivore wasn't even there. And we found that then in that case, the algal variation was really high. So in this experiment, stronger predator-consumer interactions increased stability in the algae, and that was in the omnivores and in the two species that had stronger interaction strengths between the crayfish and the snail. So that was a neat experiment because we were able to create predators, and we were able to maintain the identity of the predator, or the omnivore in this case. In the experiments that I talked about earlier, they always switch the identity of the omnivore. So in order to change how omnivorous a predator is, you often have to change the identity of the omnivore. So this was a cool example where we kept the same species, but were able to manipulate its feeding. So next, I want to talk about an experiment of omnivory in situ. So the experiments that I have just talked about earlier was a cool experiment we did, but we were doing it in a mesocosm. So what about looking at a place where an omnivore has been introduced? Again, do we find that these predictions of omnivory, do they exist in this context of omnivore introduction? So I'm going to talk to you about a place where omnivores have been introduced. So this is the shape of the Magdalen Islands. The Magdalen Islands are it's a territory actually of Quebec. So if you don't know where it is, if you drive through New Brunswick and then across the bridge to PEI, and then you take a 5-hour ferry, you get to the Magdalen Islands. So you get off the ferry and it says, welcome to Quebec, and you get confused. And everyone there has actually a very regional accent as well. So, and great windsurfing and really cool sand dunes. So if you ever want to visit and continue to speak French, that's the place to go. So what else besides great sand dunes and windsurfing does the Magdalen Islands have? They have mussel aquaculture. So in a couple of the bays, the bay that I looked at was the Have-a-Maison enclave here, and what they do there is they have mussel aquaculture on long lines. So the way that mussels generally grow is that they are benthic organisms. If you've ever gone snorkeling in one of the lakes around here, you might see some mussels down in the benthos. That's normally where they're found naturally. But for mussel aquaculture, and for them to grow them efficiently, they grow them on these long lines. So these are all mussels on these lines that are suspended from the benthos up to sort of like the surface of the bay. And so what happens when you manipulate the distribution of mussels is that now the mussels are no longer in the benthos, they're in the pelagic zone, and they have now access to novel resources that they didn't before in the benthos, including zooplankton. And zooplankton are competitors of mussels. So zooplankton eat algae, mussels eat algae, but now that mussels are in the pelagic zone, mussels can also eat zooplankton because their inhalant siphon is big enough to accept zooplankton particles. So they just kind of like filter, and if you're a small little zooplankton and you're the right size, they're going to eat you. They don't really care. So essentially what that builds is this, again, omnivory configuration where you've got a predator that consumes a consumer, that consumer has a resource, and then that omnivore also eats that common resource. So that module that I've been talking about throughout the talk. And so what are the effects then of introducing this omnivore? Does it mean that you've got more persistence? I mean, going in you wouldn't think so, because all of a sudden, if you were a zooplankton, you are fine in the pelagic zone, only having to compete with other zooplankton, and all of a sudden, you've got this high density of mussels that are not only they're only competing with you for this common resource that you didn't have to compete with before, but also are eating you. And so you would expect that this configuration actually should be really unstable. You shouldn't find that any zooplankton, because they have this double jeopardy of predation and competition. So we went out there to look at what the densities of the zooplankton were in farm and reference sites. So the farm sites are where we have these long lines of mussels, and in the reference sites there's no mussels at all. This is a non-metric multidimensional scaling plot